Okay, this is going to turn out to be my final study guide. I'm running out of time here, so away we go. Be able to identify the different types of blood vessels and how they function to transport blood throughout the body. So components of perfusion, these are all going to be critical, right? Cardiovascular system circulates the blood throughout the body, oxygen, nutrients, removal of waste products. Perfusion is the delivery of blood per unit time, per gram of tissue. That's the calculation that we're going to want to remember. So milliliters per minute per gram of tissue. And then this obviously helps maintain healthy body cells. Requires continual pumping of the heart, open, unblocked vessels. The next thing that it's asking for is let's be able to identify the 4 different chambers of the heart and their respective locations, right? So, so here's the breakdown that we're going to want to keep in mind. Right atrium receives deoxygenated blood from the body. Right ventricle pumps deoxygenated blood from to the lungs, rather. So right side, atrium and ventricle, are the deoxygenated side. And remember, when you're doing heart placements, everything's goofy foot, right? Anatomically, so it's backwards. Right ventricle and right atrium are looking at on page left, and left atrium, left ventricle. So left side receives the oxygenated blood from the lungs, and the left ventricle pumps oxygenated blood to the body. So again, right side deoxygenated, left side oxygenated. Ventricles are the pumping systems. Atriums are the receiving. We are going to go through the great vessels now. The actual question is, what are the main great vessels discussed, and what is their general function in the body? So, great vessels are the large veins and arteries that are directly attached to the heart. On the right side is the vena cava, SVC, superior vena cava, and inferior vena cava, IVC. They drain deoxygenated blood into the right atrium. Remember, again, right side, deoxygenated. The pulmonary trunk is a great vessel that is also on the right side. This receives deoxygenated blood pumped from the right ventricle. On the left side, you have the pulmonary veins. They drain oxygenated blood into the left atrium. And also on the left side is the aorta. This receives the oxygenated blood pumped from the left ventricle. To create some quick kind of visual reference, the aorta is the large donut kind of thing that wraps over the top, right? Very thick, lots of branches, 3 main branches going across the top. The superior and inferior vena cava will generally be the straight hose that is on the backside, if you will. And then the pulmonary trunk is the thing that pulls through the middle hole of that aorta. And then that is where you'll see the pulmonary veins, 4 of them, exiting, 2 on each side, right? So that's kind of the look of this. We're going to continue to go through the specific functions of the great vessels. So on the right side, vena cava, the large veins, they drain oxygen-poor blood into the right atrium. Superior vena cava, head, neck, upper limbs, superior trunk region. The inferior vena cava services lower limbs, inferior trunk regions. This is the body's largest vein, the vena cava. And then also again on the right side is the pulmonary trunk. This is a short artery that receives oxygen-poor blood from the right ventricle and sends it to the lungs. So now moving to the left side, you'll have the pulmonary veins. Their purpose is to drain oxygen-rich blood from the lungs into the left atrium. The aorta is what is receiving oxygen-rich blood from the left ventricle, and it transports it throughout the body. Here are the valve components. AV valves, atrioventricular, located between the atrium and the ventricle. You'll have semilunar valves. This is going to be the boundary between ventricle and its associated arterial trunk. And then valve function, allow unidirectional blood flow and prevent backflow. On the right side, you'll have right AV valve, also called the tricuspid valve. Between the right atrium and ventricle, and the pulmonary semilunar valve will be between right ventricle and pulmonary trunk. To create the visual, when you're looking at the heart, on the upper right is where you're going to find the mitral valve and the aortic valve. Aortic valve will be very round in nature and tucked into like the top left of the right side. So the heart as a whole, it's quite central. And the mitral valve will be right next to it, basically. The pulmonic valve, this will appear to be in left center. And it has kind of like the double umbrella shape handle. It's like a traditional U. When pressure is high, the valve is open and then the U's come down to touch each other and close. That's the pulmonic valve. And the tricuspid valve will be lower left. Now moving on to the circulation systems. General pathway of pulmonary circulation. It shows your pulmonary vein, and keep in mind this is the oxygen-rich side. So oxygen-rich exiting lungs, fills aorta, the main artery, left atrium of the heart, oxygen-rich again, along with the left ventricle, is taking all of this oxygen-rich blood, and it gets pumped by the ventricle, obviously, and then goes through systemic circulation where it makes its exchange with the capillaries. Everything takes the oxygen that it needs, picks up the carbon dioxide, and then up it goes. So pulmonary circulation, this is just dealing with lung exchange from lung to heart. So let's be very concise about when they differentiate between pulmonary and systemic, okay? So to be very clear and concise, this is heart of the right side to the lungs to the heart on the left side. Pulmonary circulation, movement of blood to and from the lungs. Oxygen poor blood transported from the right side of the heart to the lungs via blood vessels. The gas exchange occurs within the lungs between alveoli and capillaries. Oxygen moves into the blood from alveoli. Carbon dioxide moves into the alveoli from the blood. Oxygen-rich blood is then transported to the left side of the heart via the blood vessels. Systemic circulation goes from heart, the left side, to the systemic cells, and then to the heart on the right side. So it's movement of blood to and from systemic cells, oxygen-rich blood transported from the left side of the heart to the systemic cells via blood vessels. The gas exchange occurs at the systemic cells. Oxygen from the blood flows into the cells. Carbon dioxide from systemic cells flows back into the blood. Oxygen-poor blood is transported to the right side of the heart. Okay, this is the portion of the exam where it wants us to understand the function of the pericardial sac and what tissues it's made up of. Understand the function of sulci in the heart and be able to identify the different types. Be able to identify different layers of the heart wall and the main functions of each layer. And then we'll catch our breath after we go through that. That's what transitions into sarcomeres. So we're going to deal with a lot of the tissues, location, position, things of that nature to be able to exhibit an understanding of where the heart is, what it's made up of, things of that nature. So fibrous pericardium, this is what encloses the heart without attaching to it. It's dense, irregular connective tissue attached to the base of the arterial trunks, pulmonary trunk, and the aorta. The parietal layer of the serous pericardium, this is number 2, adheres to the inner surface of the fibrous pericardium. It's made up of simple squamous epithelium and underlying delicate layer of connective tissue. So the visual on this, it's trying to convey, a very good example is the pulmonary trunk area of the heart, so upper right, also up against an atrium, catching chamber, so left atrium, and it's showing the fibrous pericardium is the pericardial sac. So parietal layer of serous pericardium, that's what is within the sac. So visceral layer of the serous pericardium directly adheres to the heart, and this is through simple squamous epithelium and an underlying delicate layer of connective tissue. The serous layers, they're continuous with one another. and they're separated by the pericardial cavity. And the pericardial sac is a loose-fitting bag, if you will, that encloses the heart. It's composed of the fibrous pericardium and the parietal layer of the serous pericardium. Okay, we're going to go through the sulci. These are external grooves on the heart. They contain the coronary blood vessels to supply blood to the heart wall. They mark the separation between ventricles and atria. The types are coronary, atrioventricular sulcis. They separate the atria and the ventricles, anterior and posterior interventricular sulcus, yes? So separating all four chambers. The coronary sulcus, it has an external groove. It separates the atria and the ventricles. So it's the jagged line that basically separates top and bottom, as if the heart were kind of looking like the Indian subcontinent. So what they call the base of the heart, which is toward the top, and the apex of the heart, which is the bottom. And you'll remember one of these images shows the very apex of the heart is generally indicated all the way to the bottom right. Coronary sulcus, right coronary artery in the coronary sulcus. So I'm looking at an image that shows it basically on the right side and the left side. So you have a very good idea of it's basically the main river flows, if you will, that separate left and right, and then it gets jagged and branches off. Very large external groove. That's going to be one of the mainstays. Intraventricular sulci. This is the external groove between the actual ventricles. So when it's talking about intraventricular sulci, it's mainly exhibited on the left side for some reason, anterior and posterior. These are basically the main branches of the river. So yes, again, these are just the main river branches coming off of the largest sulci. Heart wall layers. Okay, we have to know these layers. This is the section right before it starts getting into the sarcomeres. So the external epicardium contains visceral pericardium, simple squamous tissue, and an underlying layer of connective tissue. Thickens with adipose tissue with aging. So this is going to be a very thick cell structure, simple squamous epithelium on the very outside, so a thin layer. Epicardium is generally exhibited as yellow to indicate the areolar connective tissue and adipose connective tissue. The middle myocardium, the very thick layer, generates heart contractions. It's a cardiac muscle tissue, and it's the thickest layer. The internal endocardium covers the internal surface of the heart and the external surface of the heart valves. It's a simple squamous tissue and underlying layer of connective tissue. So that's the endocardium. Okay, this section's going to go pretty fast. This is regarding sarcomeres and cardiac muscle. So myosin thick, actin thin, I bands, they contain only actin. They shorten during contractions. A bands are where actin thin, myosin thick overlap. Z disc, where actin thin connects and creates a unit. The H zone contains only myosin thick. So this is the diagram of the person trying to hold up two walls, with the Z bands being the walls, the actin being like the resistance bands, if you will, connecting to our exerciser's body. The M in the middle would be the human body in this diagram, the thick filaments. So if you can remember this picture in your mind, it will kind of give you a map of the image that you want to go through the sarcomere construction. So the Z bands would be the outside walls, actin being the resistance bands, and then the myosin, the thick filaments, being the body trying to hold up the walls. We're going to go through the electrical signals of the heart and what's going on there. So this is asking us to exhibit some kind of knowledge of sinoatrial node, the pacemaker of the heart, if you will. This is the small round node that's toward, that's in the right atrium. So again, backwards, right atrium, left side, top left. The atrioventricular node, the AV node, is the next one down under the SA node. And see, then that will transition to a bundle. The bundle goes down through the middle and branches off left and right to all the Purkinje fibers. So the actual structures, SA node, sinoatrial, are the pacemakers of the cell. They initiate heartbeat by signaling the atria to contract. Atrioventricular AV node disconnects electrical systems of the atria and ventricles. That's the lower node that actually has a bundled neurons, if you will. It looks like a river on the page. It to flow away. So the AV node is the node that's under the SA node that connects to the rest of the system. The atrioventricular node transitions into the rest of the bundle. The AV bundle carries electrical signals from the AV node to the bundle branches, and it divides into the left and right, and then the Purkinje fibers extend from the bundle branches and contract the actual ventricles, again, the pumping mechanism. So the major events are step 1, SA node initiates action potential and spreads throughout the conduction system. Step 2, the spread of action potential gets released throughout the system. The cardiac muscle cells, action potential is initiated at the plasma membrane of the muscle cells, and the sarcomeres then contract. So when the SA node is at rest, this gets into the regulatory, the sodium leak channels, the potassium leak channels, sodium potassium pumps, and what those gradients are. So there's more sodium outside the cell and more potassium inside the cell. And then the calcium, they establish the concentration gradient. It starts getting into action potentials. So pacemaker potential, the depolarization, repolarization, and the thresholds. This is where we're going to impress the entire cardiac cycle. So step 1, atrial relaxation or ventricular filling. So step 1, atriums relax and ventricles fill. Step 2, atrial contraction and ventricles continue to fill. At which point, both ventricles will reach their maximum filling capacity. So step 3 is the isovolumic contraction. 4 is the ventricular ejection. And 5 and final is the isovolumic relaxation. Okay, so this is ECG, EKG, the electrocardiogram. What information does it provide? It monitors the electrical activity within the heart. The electrodes get attached to the skin with a collection of signals, get charted. P waves are atrial depolarization, and they originate in the SA node. Secondly is the QRS complex. This is recording ventricular depolarization. P waves generally 0.08 to 0.1 of a second. QRS complex, the ventricular depolarization is occurring between 0.06 and 0.1 of a second. There's a larger signal due to the size of the ventricles. And then step 3 is telling us the T waves, which is the ventricular repolarization. Now we're moving on to cardiac cycle and specific pressure events. So we're going to remember that systole is the contraction of the heart chamber, and diastole is the relaxation of the heart chamber. So the systole or the contraction is the top number, and the relaxation event is the bottom number. Alternating contraction and relaxation creates pressure changes within the heart chambers, and that equates to pressure gradient, creates an unidirectional movement of blood. This is what creates the opening and closing of valves to create backflow. So we're running short on time, and we're going to wrap this up. It makes a really big deal at the end about systolic and diastolic pressure, right? The normal number that you see, 120 over 80, blah, blah, blah. So it also wants to remember, so systolic minus diastolic, and this is, this creates the final number, so don't forget that math. And then pulse pressure, I'm almost certain that's what the math formula is. Systolic minus the diastolic creates the pulse pressure. And then just to be very clear on the main events of the cardiac cycle, step 1, atrial relaxation and ventricular filling. Step 2 is the atrial contraction, at which point ventricular filling continues. Step 3 is the isovolumic contraction. Step 4 is ventricular ejection. And step 5 is the isovolumic relaxation. The end, it's wrapping up on like the liquid pressure. It wants us to know those general numbers of blood pressure and things of that nature. So it's ending with the math of the end diastolic volume, which is generally about 130 milliliters. You subtract that by the stroke volume, which is generally about 70 milliliters, and that will equal the end of the systolic volume, so the ESV. So it's EDV minus SV will equal ESV. Okay, we're moving on. Arteries transport blood away from the heart. Veins transport blood toward the heart. Capillaries is where the exchange occurs between arteries and veins. Tunics, tunica intima, innermost layer. It has endothelium, provides a smooth surface as blood moves through the lumen. Releases substances that regulate the contraction and relaxation of the smooth muscle in the tunica media. Tunica media, the middle layers, smooth muscle cells arranged in circular layers. Contraction results in vasoconstriction. Relaxation results in vasodilation. So vasoconstriction, squeezing, contracting, smaller, increasing blood pressure. Relaxation is the widening of the blood vessel lumen, so the pathway is getting larger. Tunica externa is the outermost layer, contains connective tissue. So we're getting into the portion where it is just going to pound home all of the differences between arteries and veins. So this is the category of lumen diameter. Arteries are narrower than vein lumen. Veins are wider than artery lumen. The general wall thickness. Arteries' walls are thicker than a vein. Veins thinner than an artery. The cross-sectional shape. Arteries retain, the cross-sectional shape is retained even without blood in the vessel. Veins, cross-sectional shape, tends to flatten out or collapse without blood in the vessel. The thickest tunic of the artery is the tunica media. In a vein, it's the tunica externa. Elastic and collagen fiber differences in tunics. Arteries have much more than a vein. Veins have less than an artery. Arteries don't have valves. Valves are present in most veins. Blood pressure range in arteries is higher than in veins, generally 100 millimeters Hg. In larger arteries to 40 millimeters Hg in smaller arterioles. Blood flow. Arteries transport blood away from the heart to the body. Veins transport blood from the body to the heart. Blood oxygen levels. Systemic arteries transport blood high in oxygen. Pulmonary arteries transport blood that is low in oxygen. Systemic veins transport blood low in oxygen, and pulmonary veins transport blood high in oxygen. So moving on to some of the main characteristics. Artery characteristics: lumen diameter narrow. Vessel walls are thick. Thickest tunic is the media. Blood pressure range is higher. Arteries are transporting blood away from the heart to the body. Systemic arteries transport oxygen-rich blood. Pulmonary arteries transport oxygen-poor blood. Vein characteristics: lumen diameter is wider, vessel walls are thin. The thickest tunic is the tunica externa. Blood pressure range is lower. Transports blood from the body toward the heart, veins do. And systemic veins transport oxygen-poor blood. Pulmonary veins transport oxygen-rich blood. Elastic arteries, these are the largest. They conduct blood from the heart to the smaller arteries. The muscular arteries, the distributing arteries. Let's not forget, elastic are also known as the conducting arteries. Muscular arteries are distributing. They're medium-sized. They distribute blood to specific body regions and organs. And then the arterioles, smallest. And they may have all three tunics or down to only one tunica intima. And they're responsible for vasomotor tone. Let's stay aware of the types of capillaries. Continuous capillaries, they're the most common type. They do have an intracellular cleft, gaps between endothelial cells. They allow movement of small substances, water, glucose, amino acids, ions, stuff like that. They prevent the movement of larger substances, so they basically house all of our formed elements, right? And then we have the fenestrated capillaries. Their composition is relatively the same as continuous, but they contain pores, the fenestrations, extremely thin regions of the endothelial cells. This allows for the movement of the small plasma proteins, prevent movement of formed elements. You have these the fenestrations for the pinocytic vesicles and the erythrocytes. And then we move on to the sinusoids, which contain very incomplete linings, so really huge openings and gaps and fenestrations. And this is for the transport of larger substances, the formed elements, plasma proteins, to move between the blood and tissues found in red bone marrow, liver, some endocrine glands, things of that nature. So let's try and keep in mind a general overview, because these kind of tricky questions always pop up. Comparison of vessels. Veins merge and drain into larger vessels. Venules are the smallest veins, and they're only 8 to 100 the nanometers. The venules merge to form veins. It's considered a vein when its diameter becomes larger than 100. The function: blood reservoirs, low blood pressure. Let's see. It contains 55% of the body's blood at rest. Blood is shifted from venous reservoirs into circulation via vasoconstriction with increased physical exertion. This is where the simple pathway of blood vessels gets kind of breezed over. And the function of this is to go from artery to capillary bed to the veins to the heart. One major artery delivers blood to the target organ, and it branches into the smaller arteries, with the smallest being the arterioles. They feed into a single capillary bed, and then the venule drains the blood from the capillaries and merges to form a major vein. That completes the cycle of what is known as the simple pathway. And then we get into the other alternative pathways, anastomosis and the other portal systems. They differ in the number of arteries and capillary beds and veins that are involved that serve an organ or body region. So there are multiples, right? Anastomosis, the joining of the blood vessels, if the alternative pathway of a vessel is blocked. So there are three types that they refer to. Arterial are 2+ converging arteries to supply the same body region. This happens with the coronary arteries. The venous are 2+ veins that drain the same body region. This happens in the brachial veins. Arterial venous, or the shunt process, transports blood directly from an artery into a vein, and this bypasses the capillary beds present in your fingers, your toes, your palms, ears. This is triggered in hypothermic conditions, and what makes you vulnerable to frostbite. So this is what can trigger the arterial venous or the shunt response. So now we're getting into the portal system. Blood flows through 2 capillary beds. So you'll have that typical wiry meshwork, kind of like a spider web on acid on each side. The portal vein delivers blood to another organ before sending the blood back to the heart. So the pathway of this goes from artery to capillary bed to portal vein, back to a capillary bed, and then back to a vein. The example of this is the hepatic portal system, or the transport of blood from the digestive tract to the liver. This is where it wants to make the transition into blood flow velocity. The arteries, veins have a smaller cross-sectional area, so they're relatively fast velocity of blood flow, yes. The capillaries have a larger cross-sectional area, so they have a relatively slow velocity of blood flow. This allows for the sufficient time for exchange of gases, nutrients, waste, and hormones that need to take place between the tissue and the blood. So they're going through actual capillary exchanges. So diffusion is when substances move from higher to lower concentrations, like systemic capillaries. Oxygen, hormones, nutrients move from blood into the tissue cells. Carbon dioxide and waste move from the tissue back into the blood. Small solutes can be exchanged. Glucose, ions, oxygen, carbon dioxide, and fluids diffuse along with some endothelial cells. And then larger solutes, proteins, pass through the fenestrations and sinusoid gaps. Secondly, the other process next from diffusion is the actual vesicular transport. So this is where the endothelial cells to form vesicles, which are transported on the other side of the cell and released via exocytosis, movement from the blood into interstitial fluids or vice versa. This happens with relatively large solutes, the example being insulin. And then from diffusion to vesicular transport, we move into bulk flow exchange processes. And this is the movement of large amounts of fluids along with their dissolved substances in one direction down a pressure gradient. Okay, capillary exchange. We just got into bulk flow. Let's see. Reabsorption, fluid moves back into the blood, the venous end of the capillary. 85% of fluid that passes into the interstitial fluid from the blood. The remaining is absorbed by the lymphatic system. So this is taking place at the venous end of the capillary. The filtration is what is taking place at the arterial end. The fluid moves out of the blood through capillary openings, the intracellular clefts and fenestrations, and the arterial end of the capillary is what's doing all the filtering of fluids and small solutes. It blocks formed elements and proteins. So now we're getting into the actual pressures that relate to and cause these interactions for the capillary exchange. So these are all the different pressures associated with fluid regulation in the vessels, and their actual abbreviations and how they're going to be referred to. So HP is hydrostatic pressure. This is the physical force exerted by a fluid on a structure. Blood pressure is the hydrostatic pressure. This is HPB, blood pressing on the internal surface of the vessel wall. The interstitial fluid pressure, which will be in the parentheses HPIF, interstitial fluid, okay? So interstitial fluid pressing on the external surface of a blood vessel. And then we get into the colloid osmotic pressure. This is the pull of water back into the tissue by the tissue's colloid protein concentration. This will be listed as the blood COP, so COPB. Again, colloid osmotic pressure for the blood protein, COPB. This force draws the fluid back into the blood due to blood proteins. Interstitial again, IF, interstitial COP. The force draws fluid into interstitial fluid due to the two proteins in the interstitial fluid. This is not very common. So capillary exchange, net filtration pressure is the other thing that's going to come up. The difference between the net hydrostatic pressure, HP, B, and IF, and the net colloid osmotic pressure, the COP B, COP IF. This is also the variation of Starling's law. Hydrostatic and osmotic forces, they drive filtration and reabsorption of materials across the capillary walls. So you'll see this equation as NFP, net filtration pressure, equals the algebraic math of HPB minus HPIF, close parentheses, minus the colloid osmotic pressure, COPB minus COPIF, close parentheses. Okay, we're going to breeze through local blood flow. Blood delivered locally to capillaries of a specific tissue. This is signified as milliliters per minute. Directed to tissues where blood is most needed. It maintains perfusion. The amount delivered varies depending on degree of vascularization and angiogenesis, myogenic response, local regulation, and total blood flow. The degree of vascularization, is the extent of blood vessel distribution within a tissue. This determines the potential ability of blood delivery. So very metabolically active organs tend to be highly vascularized with large amounts of blood delivered, and less metabolically active organs have little need for vascularization and limited amounts of blood delivery. Angiogenesis is the formation of new blood vessels and tissues. Changes the amount of vascularization in a tissue over time. It provides adequate perfusion during anatomical changes. So an example of this would be skeletal muscle in response to aerobic conditioning. Regression would be return to the previous blood vessel state, and an individual who is physically active becomes sedentary or in fat loss. The myogenic response is a contraction, relaxation of the smooth muscle within blood vessels. This is response to changes in the stretch of the blood vessel wall, and this is to maintain constant blood flow. Myogenic response is an increase in systemic blood pressure, is additional blood volume enters the vessels. It stretches the smooth muscle cells into the vessel wall. This causes contraction and vasoconstriction. A decrease in systemic blood pressure, lower blood volume, enters the vessels. This decreased the stretch of smooth muscle cells in the vessel wall, and this causes relaxation and vasodilation. So local regulation, blood flow is continuously regulated to respond to changes in metabolic activities. It's auto-regulation initiated by inadequate perfusion, nutrient oxygen levels decline, increase in waste, things of that nature. Altered levels act as vasodilators. Vasoconstriction occurs as level normalize, and it's a negative feedback loop. So vasodilation, increase in blood flow, vasoconstriction, lower blood flow. And then on blood flow, total blood flow is the amount of transported throughout the entire vasculature, so liters per minute. Total blood flow is equal to cardiac output. Increased cardio output increases total blood flow. Decreased cardio output decreases total blood flow. So blood flow resistance principles: amount of friction the blood experiences as it's transported through the vessels influences total blood flow. So friction produced by contact between blood and the vessel wall, yes? Factors affecting resistance: blood viscosity, how thick is it? Lower viscosity, it has less resistance, and vice versa. The vessel length, the longer the vessel, the increase the friction. Vessel radius, the lumen radius changes the vessel diameter. Blood flows the fastest in the center of the vessel lumen. So laminar flow, like how water flows in a river. If you increase the diameter, you're increasing overall blood flow. Total blood flow, the amount of blood moving through the vascular per unit time. Blood pressure gradients have a direct relationship. Increased cardio output increases the pressure gradient. Resistance is an inverse relationship. Increased resistance will decrease blood flow. So now we're going through the regulation of blood pressure. So blood pressure high enough to propel blood and maintain adequate perfusion, not too high to cause blood vessel damage, regulating cardiac output, resistance, blood volume. Regulate the regulation of the system, nervous system, short-term mechanics, and the endocrine system is the long-term mechanisms. So you have neural regulation, short-term regulation of blood pressure. These are the autonomic reflexes within the medulla oblongata. The cardiac center regulates heart activity and cardiac output. The vasomotor center is the degree of vasoconstriction and vasodilation of blood vessels and their resistance. The cardiac center is regulating the heart activity, so cardiac output, cardio acceleratory center. The sympathetic division, SA and AV nodes, that's who it's communicating to the heart. Increases the heart rate, larger cardio output. Cardio inhibitory center, which is parasympathetic division, SA and AV nodes. This is to decrease heart rate and have a smaller cardio output. Neuroregulation, short-term regulation of blood pressure. The receptors are smooth muscles, receptors specific to any location. Alpha-1, they're going to hammer home alpha-1 receptors. This is respond to norepinephrine. and vasoconstriction of the vessels. So alpha 1, vasoconstriction. Alpha 1 receptor, vasoconstriction of the vessels. Beta 2 receptors. This is vasodilation. Beta 2 dilation. Beta 2 dilation. Respond to epinephrine. The other receptors are baroreceptors, sensory nerve endings that respond to vessel wall stretch. The baroreceptor reflexes respond to an increase or decrease in blood pressure. And then the chemoreceptors regulate respiration and secondary responses in blood pressure regulation, like peripheral chemoreceptors in the aorta and carotid arteries. The other higher brain centers, this may come up, the hypothalamus can increase cardio output and resistance. Examples, body temperature, fight or flight responses, things of that nature. And then the limbic system alters blood pressure in response to emotions and emotional memories. Okay, now we're going to close out with the hormones, understanding the processes of the renin angiotensin system, as well as the enzymes and the hormones that are involved with it. So the hormones, angiotensin 2, aldosterone, and ADH. Step 1 is the kidney receptors detect low blood pressure and are stimulated by the sympathetic division. Renin is released at this point by the kidney. Renin converts to angiotensinogen into angiotensin 1. 3 is the conversion. ACE converts angiotensin 1 into angiotensin 2. And then angiotensin 2, step 4, increases blood pressure by causing vasoconstriction, stimulating your thirst center, and decreasing urine formation. So hormonal regulation is long-term regulation of blood pressure. The renin-angiotensin system, the liver produces angiotensinogen. It's released into the blood. It's an inactive hormone. The kidneys respond to low blood pressure or sympathetic stimulation. They release renin into the blood. It's an enzyme that converts angiotensinogen into 1 and 2. The capillary endothelium, this is the conversion process. ACE, you'll see that over and over again. ACE, angiotensin converting enzyme. This converts angiotensin 1 to 2. So angiotensin 2 increases blood pressure, causing vasoconstriction in the vessels. This increases blood volume. It stimulates the thirst center to increase fluid intake. And it releases aldosterone and ADH, decreases urine formation.